Okay, good morning, everybody. Can we get started? All right. So I think last class we started talking about the regulation, right? The two important pathways. Oh, sorry. So the two pathways, right? Glycolysis and gluconeogenesis. And I think we are talking about two key enzymes and catalyze the first reaction in the glycolysis and the last reaction in gluconeogenesis, right? The glucose converted to glucose 6-phosphate or vice versa in the gluconeogenesis. So glucokinase and the glucose 6-phosphatase, right? So I think we finished this slide. And the next pair of the enzyme in both glycolysis and gluconeogenesis are the phosphofructose 1 kinase, right, which is a key enzyme in the glycolysis pathways and catalyze conversion of fructose 6-phosphate to fructose 1,6-biphosphate. And the other enzyme, which is key enzyme in gluconeogenesis, is fructose 1,6-biphosphatase, right? And now we're going to talk about how this pair of enzymes in two different pathways are regulated. And this pair of enzyme regulation is kind of unique, or not, kind of special. That we wanted to introduce one enzyme that regulates this pair of the enzyme, which is called a bifunctional enzyme. So bifunctional enzyme basically contains the two enzymatic activities, right? Two enzymes form a complex we call a bifunctional enzyme. So bifunctional enzyme contains phosphofructose 2 kinase and fructose 2,6-biphosphatase. If you probably already noticed, right? These two enzymes is different from the key enzyme in we just mentioned in here, right? This key enzyme is fructose 1,6-biphosphatase, right? Or phosphofructose 1 kinase, but the bifunctional enzymes actually two, right? Phosphofructose 2 kinase. That means this enzyme catalyzes a different reactions, right, than the key enzyme. So and fructose 2,6-phosphate is a similar, enzyme that catalyzes different reactions. And this bifunctional enzyme, the main function of the bifunctional enzyme is to catalyze the formation of fructose 2,6-biphosphate, right? This is the very important intermediate. This intermediate that direct regulates the two key enzymes that we just described in the last slide, right, in glycolysis and gluconeogenesis activities. So these are the two key enzymes in glycolysis or gluconeogenesis, right? We just talk about it. And this key intermediates directly regulates these two key enzymes in glycolysis and gluconeogenesis, right? So try to remember the order, how the this two key enzymes in glycolysis and gluconeogenesis regulated. And basically it's regulated by bifunctional enzyme, but not directly. So bifunctional enzyme indirectly through regulate generation or production of fructose 2,6-biphosphate intermediate. Right? So this intermediate concentration then directly regulates the key enzyme activities. Does that make sense? The order, right? So now let's talk about this key intermediates, fructose 2,6-biphosphate. So this intermediate is a synthesized and broken down by bifunctional enzyme, right? I just described it in the last slide. And this intermediate is not directly involved in the glycolytic pathway. That means it's not a direct intermediate of a glycolysis pathway, right? Does that make sense? And this intermediate concentration can be increased or decreased, or in other words, can be regulated by insulin or glucagon levels, right? And through regulating bifunctional enzyme. And in the fed condition, when glucagon levels are decreased, in the fasted condition, when glucagon levels or epinephrine levels are increased, by this substrate levels or this intermediate levels are low, and there's the key enzyme activities in the gluconeogenic pathways is increased, so then can lead into gluconeogenesis. Right? That kind of confusing. Okay, let's go to the more details, so hopefully it will help you to understand better. Now let's talk about the production of this specific intermediates, fructose 2,6-biphosphate. Again, I mentioned, right, this synthesis or breakdown of this specific intermediates is controlled or regulated by bifunctional enzyme. So left side is the reactions that is for the synthesis of fructose 2,6-biphosphate, and right side is the breakdown of fructose 2,6-biphosphate, right? The reaction. So bifunctional enzyme contain these two specific enzymes, and each specific enzyme catalyzes a different reaction in terms of the generation or breakdown of fructose 2,6-biphosphate. So for example, one of the bifunctional enzyme, phosphofructose 2 kinase, and this kinase basically catalyzes the synthesis of fructose 2,6-biphosphate, right? And this fructose 2,6-biphosphate is synthesized from fructose 6-phosphate, right? And catalyzed by this enzyme, phosphate fructose 2 kinase, right? And the other bifunctional enzyme, which is fructose 2,6-biphosphatase, and this enzyme basically catalyzes the breakdown of fructose 2,6-biphosphate and to convert it to fructose 6-phosphate, right? So again, this very important intermediate, fructose 2,6-biphosphate, is the levels of this important intermediate is regulated by bifunctional enzyme. So bifunctional enzyme again has two specific enzymes. Each specific enzyme catalyzes either synthesis or breakdown of fructose 2,6-biphosphate. Right? Is that clear? Any questions? Right? So, and this is just more information about this regulation. And the bifunctional enzyme basically is the insulin and glucagon hormone is actually on the top of the bifunctional enzyme. So insulin and glucagon levels can regulate bifunctional enzyme activities. And again, also depend on which hormones, and they can regulate different enzyme in the bifunctional enzyme, right? So for example, insulin basically upregulates, means increase phosphofructose 2 kinase, but decrease fructose 2,6-biphosphatase. So the net effect of insulin will be increase one of the bifunctional enzyme activity and decrease the other bifunctional enzyme activity, right? And what does this enzyme do? Do you remember in the last slide in terms of a regulation of the production or breakdown of fructose 2,6-biphosphate? Catalyze synthesis, right? Synthesis of fructose 2,6-biphosphate. So when insulin levels increase in the fed condition, one of the bifunctional enzyme activities increase, which is phosphofructose 2 kinase, and this kinase basically regulates or catalyzes the formation of fructose 2,6-biphosphate. So basically increase fructose 2,6-biphosphate concentration, right? So in the fed condition, when insulin is increased and the fructose 2,6-biphosphate intermediate levels increase, right? And under the fasting condition, we should have increased glucagon, and glucagon regulates bifunctional enzyme that different from insulin. So basically, glucagon increase the other bifunctional enzyme activity, fructose 2,6-biphosphatase, but decrease phosphofructose 2 kinase. And we know fructose 2,6-phosphatase, its function is to break down to fructose 2,6-biphosphate, right? So it will lead to decrease this intermediate concentration in the fasted condition. Right? Is that clear? Any questions so far? All right. So now here is the more comprehensive diagram to put all things together I just described, and to understand how the second pair of a key enzyme in glycolysis and gluconeogenesis is regulated by insulin or glucagon in fed or fasting condition, right? So the top part of this slide is the condition, and the fed condition, right? And insulin level should be increased. And we just know from the last slide that insulin levels increase will regulate one of the bifunctional enzyme, right? And leads to increase this intermediate fructose 2,6-biphosphate. And this intermediate basically regulates the two key enzymes in the glycolysis, which is the left side, and gluconeogenesis, the right side, right? These are the two key enzymes in the two different metabolic pathways, right? Remember this? So when fructose 2,6-biphosphatase intermediate concentration is increased, can stimulate key enzyme in glycolytic pathway, but decrease the key enzyme in the gluconeogenesis pathway. So eventually, insulin in the fat condition, insulin will stimulate glycolysis activity and decrease gluconeogenesis activity, right? That makes sense. So just there are several layers of the regulation that regulates this pair of the key enzyme in two different pathways, right? Is that clear? Any questions? Okay, now under the fasting condition, when we have a higher glucagon levels, and glucagon can regulate the activity of bifunctional in the opposite way, right? So upregulates or increase fructose 2,6 biphosphatase, but decrease phosphofructose 2 kinase. And because of the fructose 2,6 biphosphate, usually catalyze the breakdown the intermediates. So causing the decrease of fructose 2,6 biphosphate. So this will lead to increase the key enzyme in gluconeogenesis and decrease enzyme activity in the glycolytic pathway, right? So this is the second pair of the key enzyme in both pathways and regulates by insulin. That involves the bifunctional enzyme and the key intermediates, right? Fructose 2,6 biphosphate. Right? Any questions? Okay. So now we already know the two different pairs of enzyme, right? How they regulate by hormones and substrates, right? Now the last pair of enzymes in two different glycolysis and gluconeogenesis, pyruvate kinase and actually two key enzymes in the gluconeogenesis, right? One pyruvate carboxylase and the other PEP-CK phosphoenolpyruvate carboxylase kinase, and see how this three key enzymes regulate and can control glycolysis or the gluconeogenesis. So pyruvate kinase basically kind of relatively simple. They just regulate directly by insulin or the substrate fructose 6 biphosphate. When this insulin or fructose 1,6 biphosphate levels increase, and stimulate pyruvate kinase, right, to increase glycolytic pathways. And glucagon alanine does the opposite thing. So glucagon basically is increase in fasting, right? So decrease pyruvate kinase, so decrease glycolysis activity. And PEP-CK, one of the key enzymes in the gluconeogenesis and the regulation, actually two key enzymes, right? Pyruvate carboxylase and PEP-CK. And this two key enzymes that regulate, like pyruvate carboxylase, basically is the positively regulated by acetyl-CoA levels, which means that the cell, needs more the oxaloacetate for gluconeogenesis, right? So, and PEP-CK is just simply regulated by hormones and at the gene expression level, right? And doesn't involve allosteric or covalent modification regulation, meaning like phosphorylation or some kind of other post-translation modification. So again, PEP-CK just simply regulate by hormones at the gene expression level. It can increase or decrease PEP-CK gene expression and change the levels of PEP-CK, right? I think we finished all the regulation of glycosyl gluconeogenesis. Any questions so far? Oh, okay. Okay. Now we have two more pathways of glucose metabolism left, right? Glycogen metabolism. So glycogen synthesis and glycogen breakdown, right? The two pathways and how they are regulated. So glycogen is the major storage carbohydrate in animals, and in starch is a storage carbohydrate in plant, right? Plant. So where does it occur? You already know, right? Which tissues actually does glycogen store glycogen? Liver and muscle, right? And what is the role of a glycogen? Do you guys have an idea? What is the role of a glycogen? To store the glucose and release glucose, right? For energy use during fasting. And the role of the glycogen in liver and muscle actually a little bit different. The liver, we know basically can release glucose, right, from glycogen breakdown and to raise the blood glucose level during fasting. But muscle, glycogen breakdown, the glucose usually will not released but used within the muscle cells. Do you still remember why muscle can't release the glucose? Which enzyme that is lacking that causing? Yeah, glucose 6-phosphatase, right? Glucose 6-phosphatase is not expressed in the muscle, so muscle can, have a glucose released. Okay, so there's two metabolic pathways in terms of glycogen. One is called glycogenesis, or easily just we say glycogen synthesis, right? And the other pathway is glycogenolysis, or just glycogen breakdown pathway, right? Synthesis and breakdown, be easier to remember. And so each pathway that involve a different set of enzymes. The glycogen synthesis involve the enzymes like UDP, glucose, pyrophosphorylase, and the glycogen synthesis and branching enzymes, right? So three enzymes that involve in the synthesis of glycogen, and UDP, uridine diphosphate, is also needed for glycogen synthesis, and also glycogenin. Glycogenin is a protein that also required for synthesis of glycogen. We're going to get to the detail later. And I highlighted in red, that means this is a key enzyme in the glycogen synthesis pathway, right? So glycogen synthesis pathway key enzyme is glycogen synthesis. Yes?
Why is uridine diphosphate not UDP?
Maybe I did some. I'm sorry. Yeah, you're right. I need to correct this. Sorry. It's UDP, right? Thank you for correcting this. UDP, right? So, yeah, UDP should be right? Triphosphate. So UDP, uridine diphosphate, not UTP, I'm sorry. Okay, and then the glycogen breakdown pathway that needs glycogen phosphorylase and glucan transferase and debranching enzyme. Right? Again, I highlighted red, the glycogen phosphorylase is the key enzyme in the glycogen breakdown pathway, right? So this is the sort of a structure of the glycogen. So you can see in the middle there is this represents a protein, the glycogenin, right? Glycogenin is required for glycogen synthesis, and the black dots represent the glucose molecules, right? So you can see glucose, can be formed a glucose chain and also have a branching glucose chain. Right? You have a lot of branching. So it's very complex. The structure of a glycogen, consists of many glucose molecules, and they are linked together in the linear chain and also branching chain, right? So this is a UDP glucose, you're right. So it's a UDP glucose, not a UTP again, right? So UDP is, it's uridine and also two phosphate, right, group. And the one glucose molecule is actually linked to the UDP and forming UDP glucose. And the UDP, basically, the function of UDP in glycogen synthesis is to transfer or carry glucose to the glycogen synthesized site for synthesis of glucose, right? So glucose can just freely, goes to the synthesis site, but they need a UDP, to carry to the synthesis site, right? And it's just the formation of the UDP glucose. So first of all, glucose need to be phosphorylated to glucose 6 phosphate, and then converted glucose 1 phosphate, and then glucose 1 phosphate can transfer glucose to UDP, and then forming UDP glucose. And this is an enzyme, right? UDP glucose, pyrophosphorylase catalyze this formation of a UDP glucose, right?
So is that one supposed to be UTP or DP?
Which one? Here, this one?
Yeah.
This. Sorry. Yeah, you're right. Yes, it should be the UDP. Okay. Yeah. Sorry, I'm going to collect this and send you back the revised slide. All right. Let me see. Oh, okay. So glycogenin. The glycogenin basically is a 33 kilodalton protein, right? And the function of glycogenin is to serve as a primer molecules for glycogen synthesis. So first, glucose usually binds to the UDP, right? And form UDP glucose, and then we transfer to tyrosine residues of glycogen. So first, the glucose is transferred by UDP glucose and to the tyrosine, to add it to the tyrosine residues of glycogen. Glycogen again is a protein, right? And then you add a more, glucose and to the existing glucose chain, right? And to you add a more and more glucose, right, to existing glucose. So then you extended chain, glucose chain, and until form the 8 glucose chain, right? And this glucose chain basically a linear chain, right? And connected by 1,4 linkage. And so this short chain polysaccharides, which contain the 8 glucose molecules, is called a glycogen primer. Right? Glycogen primer usually contains the glycogen and with 8 glucose attached to the glycogen, a linear chain of 8 glucose, right, attached to the glycogen. So this is a glycogen primer. So once glycogen primer is formed, then glycogen synthesis and take over the job to extend the glucose chain, and also a branching enzyme to also works and to form the branching chain polysaccharides, right? So this just sort of the process of how complex branching chain polysaccharides and glycogen molecules or structures are synthesized. Now we talk about the regulation. The pathways. The glycogen synthesis and glycogen breakdown pathway. Right? So this diagram I just put up both synthesis and breakdown together. Right? The left side from bottom to the top is a synthesis pathway, and right side from bottom to the from top to the bottom represent the breakdown pathways, right? So glycogen on the top and glucose on the bottom. So let's look at the glycogen synthesis pathway first. So glycogen basically synthesized from glucose, right? And glucose, first of all, to be converted to glucose 6-phosphate by this enzyme glucokinase, right? This is this pathway pretty much common, right, in both glycolysis and the glycogen synthesis. And glucose 6-phosphate then be converted to glucose 1-phosphate, and glucose being then transferred to UDP and form UDP glucose. And UDP glucose then carry the glucose or transfer glucose to the glycogen and forming the glycogen primer, which contain the glycogen and also 8 glucose chain, formed from the 8 glucose molecules, right? And then the glycogen synthesis continues adding the glucose to extend the glucose chain, and also branching enzymes and catalyze it, the branching polysaccharide formation. So eventually, this you go the cycles, right? You're adding more and more glucose and extending the glucose and branching the glucose chain. So eventually, you form the very complex the glycogen structure. And UDP actually recycles, right? UDP, once they finish, they carry the glucose to the glycogen and then release free UDP, and UDP being well, actually, I think the UDP is right in the reaction, from the UDP. So UDP then become converted back to UTP, and then enter the next cycle, right? And then breakdown. So during the fasting, right, we need to break down glycogen and release glucose. So glycogen, the key enzyme breakdown, this key enzyme, the glycogen phosphorylase is a key enzyme to break down glycogen. So first, two more enzymes that also involve the glycogen breakdown, the glucose transferase and debranching enzymes, and they all work together and to break down glycogen. So, and first of all, glucose from glycogen being converted to glucose 6-phosphate and then to glucose 1-phosphate and then glucose 6-phosphate. And this is a key enzyme, right? It's required for formation of free glucose, right? So, so this enzyme is very important. Without this enzyme, that glucose cannot be released from glycogen breakdown, right? Which happen or occur in the skeletal muscle. So, and also I think I mentioned the key substrate. If you still remember glucose 6-phosphate when we talk about the fructose intolerance, remember? Because of the not fructose, remember, the patient has a glucose 6-phosphate deficient, remember, right? So glucose 6-phosphate is the substrate that can inhibit the key enzyme, glycogen phosphorylase. So that means when we have this substrate accumulated, then this key enzyme will be inhibited. So that means we'll reduce the glycogen breakdown, right? So glycogen cannot be break down and accumulated. So that's the reason that we explain why in the glucose 6-phosphatase deficient patient, not actually, you have a glycogen accumulation, right, in the liver. And glucose is also regulates this key enzyme. When we have a high glucose, and it also inhibits the glycogen breakdown, which makes sense, right? During the fat condition, we have lots of glucose from the diet, so we don't need to break down glycogen, so which makes sense. The high glucose level should inhibit the glycogen breakdown pathway, right? Inhibits the key enzyme. Okay, any question so far? No? Yeah, this I just put connect this synthesis and breakdown pathway to hormone levels and to the physiologic conditions. So under the fat condition, we have higher insulin and lower glucagon, right? So insulin to glucagon levels is increased, and this will promote the pathway from bottom to the top, right? To promote the glycogen synthesis pathway. So insulin basically regulates, actually stimulates glycogen synthesis. And during the fasting condition, then the glycogen breakdown pathway is activated, right? Shall we take a break? Okay. All right, let's move on. So let's just finish the glycogen metabolism, right? The regulation. So how glycogen pathways that are regulated, and under the different physiological condition, again, the hormones change are the key, right? And the driving force for the activity, regulation of the activity. So, let's see. So the hormones, that 3 hormones regulates the glycogen activities, breakdown or synthesis, like insulin, glucagon, and epinephrine, and key enzymes that are regulated by hormones are glycogen synthesis and glycogen phosphorylase, right? One, in each key enzyme in each pathways. And intracellular second messages like cyclo-AMP, CAMP, it's important that the levels of CAMP actually change in response to the hormone change, right? So one kind of mechanism that in terms of the regulation of synthesis and breakdown is so-called cyclo-AMP dependent regulation. So for example, in the insulin, try to get all of this. Okay, so under the fat condition, right, insulin increase that can regulate cyclo-AMP levels, basically decrease cyclo-AMP levels. The reason insulin decrease cyclo-AMP level is because insulin can regulate this enzyme, cyclic nucleotide phosphodiesterase, and this enzyme basically catalyzes the formation of cyclo-AMP. So when insulin inhibits this enzyme, then the formation of cyclo-AMP is decreased. So we have a lower cyclo-AMP in the insulin, and the plus sign usually you guys know, right? It means that stimulates, right, or activates, and minus sign is like inhibits or decrease, right? So glycogen synthesis basically is regulated by cyclo-AMP levels. When cyclo-AMP levels decrease, the glycogen synthesis activities increase, right? But the glycogen phosphorylase is positively regulated by cyclo-AMP levels. So cyclo-AMP levels increase, basically stimulates glycogen phosphorylase, right? So under the fasting condition, when we have a higher glucagon or epinephrine levels, the cyclo-AMP levels increase, right? And which stimulates the glycogen phosphorylase, so stimulate glycogen breakdown pathway activity, right? So just try to remember that cyclo-AMP is kind of a connection, connect insulin or glucagon levels to the both key enzymes, right, in different pathways. So in general, under the fasting condition, or you have a higher glucagon and epinephrine, cyclo-AMP levels increase. So cyclo-AMP levels increase in the fasting condition, right, stimulated by glucagon and epinephrine. And fat condition or insulin, you just go opposite direction, right? So you just remember one condition, and it's easier to understand the opposite condition. So again, cyclo-AMP levels increase under the fasting condition by glucagon and epinephrine, and that usually will promote glycogen breakdown, right? So glycogen phosphorylase is activated or increased during the fasting and by increased cyclic AMP level. And this pretty much the same thing. I just wanted to, give you a better view how insulin regulates cyclic AMP, decrease cyclic AMP level, basically because insulin inhibits the nucleotide phosphodiesterase, right? Which this is the enzyme again that catalyzes the formation of cyclic AMP. So when this enzyme activity is decreased by insulin, then the formation of cyclic AMP decrease, according lower cyclic AMP level in the fat condition, right? Oops, sorry. So then you stimulate glycogen synthesis, but in the meantime, the glycogen phosphorylase activity decrease because of a lower cyclic AMP, right? Cyclic AMP increase basically stimulate glycogen phosphorylase. So here's just something, maybe help you to remember the high CMP level promote glycogen phosphorylase, but inhibit glycogen synthesis activity, and low CMP promotes glycogen synthesis and decrease the glycogen phosphorylase activities, right? And there's another mechanism that can also regulate the activity of the key enzyme. It's called the phosphorylation. Oh no, here not yet. Sorry. It's the glycogen synthesis and phosphorylase activities regulates usually by phosphorylation, dephosphorylation mechanism, or the cyclic AMP level we know is regulates enzyme activity, right? So downstream of the cyclic AMP is the phosphorylate or dephosphorylate. So in other words, the cyclic AMP levels increase can either dephosphorylate or phosphorylate the enzyme, then can regulate its activity, right? So, and we talked about earlier, right, the phosphorylation being just adding the phosphor group to the protein or remove the phosphor group from the protein. And the consequences of phosphorylation or dephosphorylation, in general, in most of cases, if some protein or enzyme it gets phosphorylated, the activity should be increased, right, activated. And dephosphorylation usually you decrease the activity, but it's not always the case, right? Sometimes phosphorylation, dephosphorylation can go opposite. For example, this is just a good example. The glycogen synthesis and glycogen phosphorylase, right? So glycogen synthesis, when it's phosphorylated, the activity not increase but decrease. So phosphorylated glycogen synthesis causing the inactive, and dephosphorylation of glycogen synthesis is causing active, right? So it's just different from the most of the case. And glycogen phosphorylase, it pretty much like the most of the enzymes when it gets phosphorylated and activated and dephosphorylated and inactive. It's just kind of general information. You don't have to remember details, right? And this just gives you a details and adding the more layers of regulation. We know the cyclic AMP is increased by glucagon epinephrine during fasting condition, and increased cyclo-AMP can regulate protein kinase A activities. Protein kinase A is the enzyme that catalyzes phosphorylation of the phosphorylase, right? So once phosphorylase gets phosphorylated and can be activated, and then can, what's that happen? Okay. All right. So this just give us a sort of a summary of hormonal regulation of glycogen metabolism. So under the fat and fasting condition, do you guys you have a blank or you have a field? Field, right? Okay, so that's good. Yeah, I just feel. So like a fat condition, right? I know insulin should be the main hormone that regulates glycogen pathway metabolism, and basically insulin should be stimulate glycogen synthesis, right? Because we need to store glycogen in the fat condition, and how glycogen phosphorylase is regulated under fat condition. It should be decreased, right? So again, under the fat condition, insulin could stimulate glycogen synthesis but decrease glycogen phosphorylase. So then you can promote glycogen synthesis and inhibit glycogen breakdown. And so the net result will be glycogen synthesis, right? Glycogen stored in the liver under fat condition. And fasting, we have glucagon and epinephrine levels increase, which stimulates glycogen phosphorylase and inhibits glycogen synthesis, right? So sometimes, you don't have to really remember, but just understanding, or sometimes even, thinking, okay, fasting condition, well, we need a glycogen breakdown. You need the glucose, right? So glycogen breakdown need to be activated, and glycogen synthesis need to be inhibited. So, something like this. So it should be easy to remember. So that's that. And in addition to the hormonal regulation, right, by cyclo-AMP through the cyclo-AMP mechanism, dependent mechanism, and there are other mechanisms, the regulated mechanisms called allosteric regulation. So in addition to hormones, some substrates can also regulate the the enzymes like ATP, glucose 6-phosphate, and free glucose. This can inhibits the glycogen phosphorylase, right? This is the glycogen phosphate. So ATP, glucose 6-phosphate, and free glucose basically inhibits the glycogen phosphorylase, so inhibits glycogen breakdown, which makes sense, right? When we have a high ATP, we have enough energy, or when we have a high glucose, and that means you're probably under the fat condition, right? And glucose 6-phosphate basically is converted from glucose, so it's also indicated you have a higher insulin levels or higher glucose level. So all these three conditions that indicate the fat condition, so we should inhibit glycogen phosphorylase, right? Inhibits glycogen breakdown. So that should help you remember. And then the other regulators called allosteric activator. That means that factors like 5' AMP is the important regulator, but it actually inhibits the phosphorylase activity, right? So a 5' AMP basically is present in or regulates the phosphorylase in the skeletal muscle, not in the liver, right? Because liver, the only muscle, this phosphorylase has the 5' AMP binding site. So 5' AMP can bind the binding site at the phosphorylase enzyme, so can stimulate the phosphorylase activity, and this only occurs in the muscle. And liver phosphorylase does not have a 5' binding site, so 5' AMP only regulates phosphorylase activity in skeletal muscle, but not in the liver, right? Because the enzyme, have a different characteristic. Okay, so now we talk about the hormone regulations, right? Insulin, glucagon, and epinephrine, and this regulation involves cyclo-AMP levels and causing phosphorylation, dephosphorylation of the key enzymes. And now we also talk about the allosteric regulation, the substrates. So ATP, glucose 6-phosphate, free glucose, and prime AMP, right? So again, prime AMP only regulates muscle glycogen breakdown, phosphorylase. And this is the last slide of the glycogen metabolism. Just list liver and muscle and other regulators, regulates the activities of key enzymes, and they are a little bit different, mostly are the same, but you can make a comparison. So first is the amount of a total glycogen store, percentage of tissue weight is higher in the liver, right? But the total body content of muscle is high. So like a percentage, like for example, the how much glycogen per 1 gram of a liver versus 1 gram of a muscle, usually in liver is high, right? Because it's more complex, the glycogen in the liver. But if you're thinking about the total body content, muscle definitely have a large mass, right, compared to liver. So the total, glycogen in the body muscle mass is actually high in the muscle than the liver. So it's kind of different concept. And then in terms of regulators, hormonal regulation, hormone insulin, glucagon, epinephrine, and norepinephrine. So liver, the difference is that the glucagon only regulates the liver glycogen metabolism and not muscle, right? And other regulators, the allosteric regulators, ATP, glucose 6-phosphate, and free glucose, and they regulates, liver, glycogen metabolism, and free glucose only regulates liver glycogen metabolism, but not muscle, right? And muscle, 5' AMP only regulates muscle but not liver. So I just highlighted, red, so you can make comparison. Some of the hormones or allosteric regulators only control liver glycogen metabolism but not muscle, and vice versa. Some of the factors only regulate muscle but not liver, right? And also the role of the stored glycogen is also different between muscle and the liver. This I think we know very well, right? Break down the glycogen into free glucose in the liver. Liver can do that and release glucose into circulation, but muscle cannot release glucose, right? Only break down glucose for use in the energy in the skeletal muscle, right? Because of lack of the glucose 6-phosphatase. All right, I think that's all I have for all carbohydrate metabolism. Any questions for today's lectures? No, you guys all okay? Okay, so so next Tuesday, I think the review, right? I will be here, and if you have any questions, from study guide or whatever you have, and we can talk about it. Or if you can also tell me which part you think is kind of confusing, wanted to clarify, and you can also let me know, right? Mm-hmm. Okay, and we have a quiz, right?